All right. So first of all, we will understand how these hematopoietic stem cells are responsible for the synthesis of various kinds of cells, including B and T lymphocytes. तो हम कई बार hematopoiesis cover कर चुके हैं. Hematopoiesis is the process in which a single hematopoietic stem cell, which is having the self-renewing property, is able to differentiate into different cell types, including the myeloid cells, which are neutrophils, dendritic cells, macrophages, monocyte, eosinophils, basophils, red blood cells, and platelets. Also, the same hematopoietic stem cell is responsible for the synthesis of lymphoid progenitor cell, which will develop into either into a natural killer cell, T progenitor cell finally, which will develop into the T helper cell or T cytotoxic cell. We will see how these cells are developed. B cell or a dendritic cell. All right. So these cells, B and T cells, are our prime concern today because we are going to discuss them in detail, how the maturation and differentiation of these cells is happening and which organs are responsible for their development. So we have started with the thymus. So thymus is responsible for the maturation of T cells. Okay. So we should start from the beginning. So the question is actually from where the hematopoietic stem cells are coming. So we are having three germ layers. So mesoderm is actually responsible for these hematopoietic stem cells. And we know from the previous diagram of hematopoiesis we have covered, this hematopoietic stem cell is responsible for the development, for the generation of all these different kinds of cells in the blood, which are known as white blood cells. So these lymphocytes are also one kind of white blood cells, which are making 20 to 40% of the WBCs. So this mesoderm is making this hematopoietic stem cell. Now, since we are humans, so from this period, which is around 30 days, about 1 month, to up to this time, up to here, which is around, can say roughly 60. So 1 to 2 months of our growth, the hematopoiesis or the synthesis of cells is actually taking place in the placenta, then in the liver, all right, and then in this AGM is actually for aorta, gonad, mesonephro, okay, mesonephros reason. So this is also responsible for a time period in which the hematopoiesis or the synthesis of different types of cells is actually taking place. And then about birth or afterwards, these hematopoietic stem cells are made, okay, and then these hematopoietic stem cells are responsible for the development of various different kinds of white blood cells through the process called hematopoiesis. Now, we can divide all the organs or the tissues of immune system. So the immune system consists of many different organs and tissues that are found throughout the body. All right, because these tissues and organs are present in our body. These organs can be classified into two main groups. One are known as primary lymphoid organs, providing the appropriate microenvironment for the development and maturation of lymphocytes. So the primary lymphoid organs are the organs which are providing an appropriate microenvironment so that development and maturation of lymphocyte can take place. So bone marrow is responsible for the development and maturation of B cells, B lymphocytes, whereas thymus is responsible for the maturation and development of T cells. All right. So secondary lymphoid organs trap antigen from defined tissues, vascular spaces, and are the sites where the mature lymphocytes can interact efficiently or effectively with that particular antigen. So, what is the difference in a primary lymphoid organ and a secondary lymphoid organ? Primary lymphoid organ are the organs which are providing microenvironment for the development and the maturation of the lymphocytes, whereas the secondary lymphoid organs are uh, the organs which are the sites where antigens are encountered by these lymphocytes so that they can learn how to interact with that particular antigen. All right. Now, when we will move to the development part, I will tell you most of the cells means about 95% of the cells, 95 to 97 or even 95 to 97 or 98% cells will die. They will not mature at all. Okay. Only 2 to 5% of the cells will responsible for mounting an immune response. So this is a so you can think of how strict this particular process is, because most of the cells will ultimately die. They will not mature at all. So this is a stringent procedure to select those particular cells which are beneficial to an organism, which will constitute the immune system of our body. So therefore, our immune system is highly complex system, all right. So which is actually facing uh, many steps of development, how these cells will be selected and then they will develop themselves or mature themselves. So first of all, okay, so we have covered hematopoiesis. Hematopoiesis is actually happening in the bone marrow in adults, and all right. So first of all, we will cover the okay, so one more question. Actually, one question has been asked in one of the competitive examinations. I forgot to tell you. So we have covered hematopoietic stem cells, right? So these hematopoietic stem cells are responsible for making, these are pluripotent, right? So I hope you understand the terms unipotent, multipotent, pluripotent, or totipotent, right? You know the differences, or should I explain these terms to you first? Unipotent is a type of cell which will develop into a single cell type, unipotent. Okay, it's uni for single. Multi means which can give rise to different, means two or more than two types of cells, two or three. Pluri means which can give rise to number of cells. Okay, and a toti means which can give rise to complete organism. Okay, because we are having more than 100 different types of cells. So all the cells can be developed from single hematopoietic stem cells. So hematopoietic stem cells are actually uh, kind of pluripotent cells because it will give rise to all the WBCs, all the white blood cells, by the process called hematopoiesis. So these cells, red blood cell, platelets, leukocyte, neutrophils, lymphocytes, monocyte, eosinophil, basophils, okay, macrophages, all are produced from the same hematopoietic stem cell. Now, each time when a B or T cell will start a cell cycle, two cells will be generated, out of which one is effector cell and one is memory cell. Just 1 minute. Okay, so each time whenever there is a cell cycle, when a B cell or T cell will divide, it will give rise to one effector cell, one memory cell. And the non-dividing cell is known as naive B lymphocyte, for example, which is present in its G0 state. So we know first it will move to the G naught is a state of the cell which is called resting stage. When it come into the cell cycle, first of all, in the G1 stage, the gene activation is taking place. Then the cell will move to the S phase. In the S phase, DNA synthesis happens. So the size of cell will increase. This increased sized cells are actually known as lymphoblast, blast cells. Why they're called blast cells? It does not mean that they will blast. Okay, so blast mean they are having an increased size. Then through G2, finally in the M phase, they will divide and give rise to. Okay, so this cell will move into the G naught state, or the cell cycle will repeat. And this cell will give rise to two types of cells. One is called memory cell, which are meant for next or subsequent exposure to the same pathogen, and effector cell, which is actually responsible for secreting large number of antibodies, which is also called as plasma cells. So plasma cells are produced from the B cells, and they are effector cells, means they can produce large number of antibodies. If someone asks you how many antibodies are secreted by one plasma cell, so they can secrete 100 up to 1000 antibodies per second. All right, so this is per second. This is huge. So इतने antibodies वो per second produce कर सकते. Now, actually, we are deviating from the topic. Okay, we are discussing our primary lymphoid organs, but actually, this diagram came in between. So, actually, I need to explain this to you. If you understand this, then we will move to our primary lymphoid organs. All right. Sorry. So, there are other terms also. For example, here given autologous, syngeneic, allogenic. Okay, so these terms are very common in immunology. Auto means if we are transplanting the organ or the cells from the same host, means if the recipient is the donor also, or we can say recipient and donor are same individual. We are taking the skin cells and then we are doing some skin graft, okay, in the same individual, then it is called autologous. Syngenic means they are genetically identical, means sometimes what happened, the identical twins are having same genotype. So we can transplant either these hematopoietic stem cells or the some graft or organs between two individuals which are homologous, means which are having same genes. Allogenic means the individual, the donor is an individual of the same species, but it is not having exactly same genes. Means it is human, but it is related, means not related directly to the recipient. So, what is the definition? The donor and recipient are not genetically identical, means they are both of the same species, and even some severe cases which are known as genograft or geno. Okay, so in those cases we can exchange graft between different species. So these four terms are very common. You will find them, these somewhere. All right, so first of all we will discuss actually we have covered this already. All these things we are covered. So first of all, the primary lymphoid organs. All right. So first we move to the primary lymphoid organs, which are organs of the immune system. So again, the same diagram. Okay, so it is showing thymus and bone marrow, which is actually present in our long bones, for example, femur. Okay, and we are having secondary lymphoid organ, for example, spleen, Pierce patches, lymph nodes, and these things. Now, it is already written here that this is a stringent process, and more than 95% of all the thymocytes will die by apoptosis in the thymus itself without ever reaching the maturity. Maturity se pehle hi they will die, more than 95% of the cells. Now, so thymus. Thymus is the site of T cell development and maturation. I hope I have told you several times that it is the site for development of T cells and maturation, therefore it is, these are called T cells because they are maturing in thymus. It is a flat bilobed organ situated above the heart. If you are able to observe the location of this particular organ, so it is present situated just above our heart, and it is having two lobes, therefore it is written here. It is a flat bilobed organ. It is covered with a capsule and divided into small lobules. Now you will understand what are lobules. Okay, so we are moving to the next page. It is showing the cross section of thymus. So actually, thymus is having a capsule. If you are able to see this red color, okay, so this is capsule. Capsule is the coating of the thymus, right? Then you are able to see these strands of connective tissue, which are bone-like structures. Okay, these are connective tissue. Okay, these are having blood vessels through which these cells can move to other sites and other cells can come to this particular organ. Okay, so these are having blood vessels in this connective tissue region. Okay, so this is called trabecula. Trabecula is a connective tissue. It can be divided into two regions. One is called cortex, one is called medulla. Medulla is the central part, cortex is the outer part. So actually this is present like this. This is a circular cross section. So now the location of cells, if you are able to observe, if you see in the cortex region, in the surrounding region, you are able to observe that the density of T cells, this is the thymus, so obviously the T cells are present in this particular organ. So the density of T cell is more in the periphery, and if you move to the medullary region in the central part, the density of these T cells will decrease. What it mean? It mean the T cell will move from the cortex to medullary part during maturation. And because we know more than 95% of the cells will die by the selection process, therefore only few are able to survive the stringent selection procedure, and therefore the density or the number of T cells will decrease, which are showing mature T cells in the center in the medullary region. Okay, are you able to understand or you are having some queries? So when these cells, these mature T cells or thymocytes will move towards the medullary region or the central region, they will encounter these antigen presenting cells, and therefore they will be selected. If they interact with self MSC molecules or self antigens, then they will be deleted. They will be killed. And in this process, more than 95% of the cells will be killed. Okay. We will see how it is happening. All right, so these are dendritic cells, or you can call them IDCs, interdigitating dendritic cells. Then we are having a very interesting structure also in this thymus, which are known as Hazel's corpuscles. What are the role of these Hazel corpuscles? Actually, they are concentric rings. concentric ring-like structures you are able to observe. They are made up of denatured epithelial cells. So these cells, which are called epithelial cells, okay, medullary epithelial cells and cortical epithelial cells, when they denature, when they are killed, they will develop this particular capsule or corpuscle like structure, a region which are known as which is known as Hazel's corpuscle, which were actually recognized by this particular scientist, so given this particular name. All right. Any specific other function of the Hazel's corpuscles is not discussed in the book. Only they have told that Hazel's corpuscles are found in the medulla, contain concentric layers of the degenerating epithelial cells. So these are having rings of degenerating epithelial cells in the medullary region. All right. So first of all, we will discuss the theory portion, and then I will tell you how exactly the maturation of T cells is happening. So we have covered this that the thymus is a flat bilobed organ which is present just above the heart. Each lobe is surrounded by a capsule and it is divided into the lobules. What are the lobules? So if you do not remember, these are lobules. This is a single lobule. This is a lobule. This is a lobule. So because these trabecula or the connective tissue is going deep up to the medullary region in the thymus, therefore it is dividing this organ into small lobules. All right. Which are separated by each other by strand of the connective tissue, which are called trabeculae. Each lobule is organized into the two compartments. One is called outer compartment or the cortex, densely packed with the immature T cells. So it is having immature T cells called thymocytes. All right. Whereas the inner compartment or medulla is sparsely, means it is having less density of the thymocytes, sparsely populated, having less density. All right. Both the cortex and medulla of the thymus are crisscrossed with three-dimensional stromal cells. All right. So one thing I forgot to tell you, stromal cells. We have discussed various different kinds of cells, including macrophages, dendritic cells, and epithelial cells. They are called as stromal cells in the thymus. Okay. So this is composed of epithelial cells, dendritic cells, and macrophages, which make up the framework of the organ of the thymus and contribute to the growth and maturation of the thymocytes. की growth और maturation के लिए responsible होते हैं. Many of these stromal cells interact physically with the developing thymocytes. Obviously, they need to interact with the developing thymocytes so that they can teach them that the T cell should not recognize the self antigens. Okay, nerve cells we already have covered. So they are thymic epithelial cells, which are specialized cells called nerve cells. Long membranous extensions surround as many as 50 thymocytes. So एक nerve cell near about 50 thymocytes को nourish कर सकती है, forming large multicellular complexes. All right. So everything is written. Okay, so you can learn the theory by reading this particular text from here. All right. Now, what should I tell you? So we have discussed nude mice in one class. All right. If thymus is absent or it is not developed fully, so what will happen? T cell will not, or the T cell population will decrease in the blood. Ultimately, it will lead to a disease called nude mice in the mice and Dye-George syndrome in the humans. Question already पूछा जा चुका है. So you need to remember this. This is really important. The disease due to the thymus if you remove the thymus, or if the thymus is already absent somehow, or if it is not fully developed, that can lead to a disease in humans. It is called Dijor syndrome, and mice, this is called nude mice, because the mice will be weak in morphology, and it will immunodeficient. Now, one more thing you need to remember from this particular text. So this particular diagram, all right, so this is for thymus. Thymus is not same throughout in humans. So with age, actually it is found that the size of thymus will increase initially up to the age of 10 or 12 years. Okay, there is an increase. The graph is showing the structure is correlated with this particular graph. So if this graph is growing, means the size of organ is increasing. So thymus will show the maximum size around 10 to 12 years. Then with age, it will decrease in size. And if you observe this around 60 years, we are having very rudimentary thymus. Everything is written here, and in the infant it is 70 gram. It's age dependent involution. Okay, so what is meaning involution means decrease in size, leaves an organ with an average weight of 3 gram in elderly. किसी बहुत बुजुर्ग इंसान में the size can decrease up to 3 gram. Okay, which is very small in comparison to 70 gram, which is present in the infant. All right. So before going into the bone marrow, we need to understand this in detail. Okay, so this is a structure. This is the cross section of thymus itself, which is showing that T cells, immature T cells or thymocytes are present here. Nerve cells are there, and then they are moving. They are interacting with the cortical epithelial cells, dendritic cells, or these medullary epithelial cells, and macrophages also. And finally, they are maturing. All right, so we need to understand this. So we will move to page number 305. Oh no. All right, so actually this is another chapter, okay, but it is a detailed discussion how actually the T cells are maturing. So this diagram is very, very important. Okay, so we are going to cover this. You need a break or should I continue? Actually, we are almost having 45 or 50 minutes. You need a break? Should I? Okay, so we are having a 3-minute break. We will start from 9 p.m. Okay. If you have a query from previous discussion, the dissection of thymus or the thymus part, then please ask. We will cover this development of T cells in the mice. Okay, because this is the same in humans, so we can study this particular flow chart. So other student seems not interested in immunology. Hiba is more interested. Hiba, you are a BSc student or MSc student? I do not know. Oh, you have completed MSc. So, okay, so you, which subject? Biotechnology only? Biochemistry. But biochemistry is not everywhere. You SLS student or you have done MSc from somewhere else? From where you
Sir,
have
I'm
completed?
from Kerala. Sir,
You
I'm
can.
from
Your voice is breaking. Actually, I'm not able to hear what you said. You are from?
I'm from Kerala.
Okay, you're from Kerala. Okay. Actually, I was thinking like because here we are having very limited institutions where biochemistry is provided, and actually, I have. I'm also MSc biochemistry. Then I did MTech in nanotechnology. So I have done my MSc from Dr. B.R. Ambedkar University, Agra. That's why I was asking.
Okay, sir. I have done my MSc from Kerala University.
Okay. So you did MSc. Okay, that's why. Because some of the questions you asked in previous classes, well, I was surprised because we are having some BSc students, and if some BSc student is asking some questions, so the from the level of the question, I can predict the level of the student, how much he or she is studying. All right, that's great. So you have studied immunology in your MSc also, obviously, because in biochemistry, I think there is a subject immunology. Actually, I have studied immunology in my MSc.
Yes, sir. I have studied.
Right.
But I completed my MSc in the year 2018.
Okay. So you are preparing. Start preparing now, or you are preparing from the last year itself.
Yes, sir.
Okay. All right. So we move back to the discussion. So, how this process is actually taking place, these hematopoietic stem cells, as we have discussed, these are responsible for the generation of all the cells. But here, because we are covering the thymus portion, we will only discuss about how the T cell maturation is taking place out of these hematopoietic stem cells. All right. So, we need to know this that the hematopoietic stem cells, which are in the bone marrow, giving rise to finally these T cells, right? So, first of all, in the bone marrow, the initial part when these hematopoietic precursors, or it is known as actually what you call it, T cell precursor or the T or the lymphoid progenitor cell, which are produced in the bone marrow, then these progenitor cells will move through the blood into the thymus, which is a primary lymphoid organ. All right. If you are not able to understand, I will lead you to the same diagram. Okay, so first of all, the long bones of our body, which are having bone marrow. In the bone marrow, the hematopoietic stem cells will give rise to a lymphoid progenitor cell. Those lymphoid progenitor cells through the blood will move to this particular organ called thymus, which is situated just above our heart. And here, the rest of the process will taking place. Okay, so now finally, when the T cell precursor came into the thymus, so first of all, as I showed you the diagram, it will come first, or it will be present in the cortex region first. Okay. So this cortex region is having large number of immature thymocytes. Now, the cells, why these are called immature T cells, because these are not having any. Okay, so first of all, you need to understand this: the T helper cells or the CD4 positive cells and T cytotoxic cells are known as CD8 positive cells. Okay, so that we know. So the T helper cells are actually CD4 positive cells. Whether you write positive or not, that hardly matter. Okay, so then the second type of cells are T cytotoxic cells, which are actually called CD8 positive cells. All right. So we are having two types of cells, but here why we are calling them immature T cells, because they are not having any of these receptors, either CD4 or CD8. Therefore, they are called double negative cells. All right. Early thymocyte development, or during this early thymocyte development, they are called double negative cells, which are present in the thymic cortex region, in the outer region of the cortex. And actually they are moving through four stages of development, which are called double negative stage 1, 2, 3, 4 respectively. In these stages, in the 1, 2, 3, 4, you can recognize. So how you are able to differentiate these four stages? This is a question. Okay, so if you know, please answer this. Actually, I want to ask how you will differentiate these four stages: DN1, DN2, DN3, and DN4, because they both do not have these CD4 and CD8 markers. So Hiba, only you are there in the class, so you can unmute your mic if you want to answer some question. All right, no need to write in the chat box. So we have covered that the hematopoietic stem cells are giving rise to some kind of precursor cells, which you can call DN1 cells, which are moving from bone marrow through the blood into the thymus. Bone marrow and thymus both are primary lymphoid organs, but because these are T cells, so the T cell maturation and development is actually taking place in the thymus. So it will come in the form of DN1 cell. DN1 cell will move through four stages to develop DN2, DN3, DN4, which are all double negative cells because they do not express CD4 or a CD8 molecule over their surface. Finally, the double negative cell will become double positive cell. They will start expressing both CD4 and CD8 markers and receptors over their surfaces. Finally, the positive and negative selection will be taking place in the cortex region, and it will give rise to single positive cells. So double negative, double positive, and single positive, SP. A single positive cell will develop, which is either showing the CD8 marker or CD4 markers on the surface. Respectively, they are called as TC and T helper cells. The cell will move into the medullary region where the negative selection will take place. Negative selection is a kind of procedure in which the cells, which are single positive cells, if they interact with the stromal cells in the medullary region, they will be deleted. So this negative selection means if they are showing high affinity with the self antigens or self MSC antigens, so they will be deleted because it is harmful to that particular host in if they are interacting with the same antigens. Okay, then the cells, which are single positive cells, will migrate through the blood into the secondary lymphoid organs where they will they will interact with the real antigens, which are some kind of pathogens or antigens which are coming from outside the body, which are non-self. So bone marrow is the site where B and T cells both will develop, but the maturation of T cells is actually taking place in the thymus, but the B cells are having maturation in the bone marrow itself. Arising from the lymphoid progenitor cell, immature B cell proliferate and differentiate within the bone marrow. Stromal cells, again the stromal cells are kind of supporting cells which will provide the microenvironment to the developing B cells, as we have studied in the case of thymus also. Whether the bone marrow interact directly with the B cells and secrete various cytokines that are required for the proper development of the B cells. Like thymic selection during the T cell maturation and selection process within the bone marrow eliminate the B cells with self-reactive antibody receptors. Okay, so actually what is the difference? Difference is the T cells are having T cell receptors. B cells are having receptors that are called BCR, B cell receptors, or simply we call them surface immunoglobulin or antibody molecules. So these antibody receptors are having are kind of receptors which are present over B cells. If the B cell antibodies or surface receptors are reactive, self-reactive, so those particular B cells should be eliminated by the negative selection process. This process is explained in chapter 11 in this particular book. Okay, so bone marrow is not the site of B cell development in all species. Okay, obviously it is not the site in all species. In birds, we are having another organ which is called bursa fabricius, where B cell development maturation is actually happening. Tissues associated with gut, the primary site of B cell maturation. Whereas in some other, okay, in birds, bursa fabricius, which is organ which is written here. Okay, in mammals such as primates and rodent, there is no bursa, so no single counterpart to the primary lymphoid organ. Okay, so in cattle, actually, in the sheep and cattle, the primary lymphoid tissue hosting the maturation, proliferation, differentiation B cell in the gestation is the fetal spleen. In some species, in cattle and sheep, actually, it is seen that the B cell maturation is happening in the fetal spleen, which is also known as graveyard of RBCs. All right. Later, it is the function is taken by the patches, assumed by the patch of tissues, which is embedded into the wall of the intestine, which is called Pierce patches. Okay, so later in the life, these Pierce patches will take the function, and then, sorry, I need to show you Pierce patches. All right. One more. Okay, so actually we are having some specific reasons, Pierce patches, which are embedded in the intestine. Okay, so these Pierce patches are responsible for the synthesis of in the cattle and the sheep. All right. In rabbit too uses it is actually having gut associated lymphoid tissue such as appendix, which is the primary lymphoid tissue for important steps in the proliferation and diversification of the B cells. So actually, it depends upon species in which we are talking. So different species are having different sites where the maturation and development of B cells is taking place. Okay. Now the B cell development is so there is no cross section actually, because there are different tissues and organs in the different organisms. Even appendix can also be the site for the maturation and development of B cells in the rabbit. Now the question is how these B cells are undergoing development. So we will move to the page number 251, I think. Let's try. I exactly do not know the page number. Oh my goodness. Okay, so here is the diagram how the B cells are actually undergoing various stages of development. All right. So because we know the bone marrow is the site where the development of B cells is actually taking place. So bone marrow is, if you see this particular arrow, up to here, up to this particular diagram, which are showing immature B cells or naive B cells, up to here, the development of B cells is actually taking place in the bone marrow, and this is called antigen independent phase, because here there is no antigen in this bone marrow. Real antigens are phased in the peripheral lymphoid organs, which are secondary lymphoid organs actually. So this last diagram is actually showing the part which is antigen dependent. Okay, so lymphoid stem cell or the hematopoietic stem cell will give rise to this lymphoid stem cell, which is showing the, okay, so it is that we are having three different genes, V, D, and J gene. V for diversity, J for joining. Okay, and V for variable. So these three genes are undergoing some kind of gene rearrangement. All right. So it will first give rise to pro B cell. Pro B cell is actually expressing immunoglobulin alpha and beta only. It is not expressing any antibodies, real antibodies, right? The surrogate light chains of pre-BCR, which are showing a structure of an antibody molecule, but not complete because the light chain is surrogate. It is not complete. Surrogate means it is in the infancy. It is not complete. The next stage is after pro is pre-B cell stage, in which the D. Okay, so the two genes were here, which are which are undergoing the gene rearrangements, D and J, diversity joining region. But here one more gene called V gene came, and this H is written, so it is responsible for heavy chain gene rearrangement. So the heavy chain are formed, but actually light chains are not complete. It is showing surrogate light chains in the pre-B cell stage. Finally, what will happen? The V and J gene, actually the light chain do not have this diversity gene. So it is having only two genes. One is variable, one is joining. So D gene is absent. Okay. So the rearrangement in the genes in the light chain, L4 light chain, will give rise to the complete antibody molecules over the surface of B cell. So finally, an immature B cell is formed. Immature B cell will give rise to the naive B cell. As I told you, naive B cell will divide, will undergo cell cycle, and it will give rise to the two cells, effector cell and memory cell. Finally, what will happen? The naive B cell will show IgM molecules over its surface when it will mature, or when it will face the antigen in the peripheral lymphoid organs or in secondary lymphoid organs. It will also express IgD molecules. So actually, the mature B cell. So this is a question. The last cell is a mature B cell, which is formed in the secondary lymphoid organ. If you see the differences in a naive B cell and a mature B cell, naive cell is showing only IgM over its surface, whereas the mature B cell showing IgM and IgD molecules over its cell surface, right? Then when it will encounter some antigens, or it will mount an immune response, these B cell will undergo class switching. And finally, it will also give rise to other different type of antibodies. So it is when it will encounter some antigens or when there is an immune response, class switching will result, and this class switching will lead to generation of other antibody molecules. So first of all, plasma cell will form, which is an effector cell. Plasma cells are formed. Plasma cells will secrete large number of antibody molecules, which are IgE, G, IgA. Okay, so actually it will secrete all the different types, including M and D. All right. But the mature B cell, which is having surface immunoglobulin molecules, will have two only, M and D. The plasma cell will not have surface antibodies, but it will secrete large number of all the antibodies, and this process is known class switching. Class is switched. We will also discuss all these events of rag 1 2 tdt h, these heavy chains, light chain genes, uh, d sorry all the three vdj and vnj in the case of light chain, in the chapter where we will discuss the vdj recombination. All right, so here you need to understand this. There are three genes in the heavy chain which are called vdj, and there are only two genes in the light chain which are vnj, which will be responsible for the development of these antibody molecules. Okay. And the second thing is the light chain is actually two types. Either it is kappa or lambda, whereas the heavy chains are of five different types. So all the antibody molecules, why we call them IgG, IgA, IgM, IgD, IgE, because of the heavy chain. If it is having gamma heavy chain, it is IgG. If it is having alpha A, mu M, delta D, epsilon E. So depending upon the heavy chain, we call the antibody either IgG if it is having gamma heavy chain. Okay, G for gamma. So depending upon heavy chain, we call the antibody molecules, but the light chain is also very, very important. To make a complete antibody structure, we do require both heavy as well as light chain. All right, that's all for today. If you have queries, please ask. If you do not have, feel free to write me in the WhatsApp.